Thank you, Simon. Thank you, Tom, for having me here. Great introduction from Felix, getting us ready in terms of having our corpus of studies so that we know what we want to meta-analyze and we're ready for the next step. And then thank you to Tom for providing the background in terms of the range of different methodologies that we have available to us. This is going to be intended more as okay, so I have my coordinates and what do I do next? This is more of a practical, let's walk you through some of the options that you'll encounter. Tal's going to handle the MKDA and the neurosynth framework, but I'll be doing the ALE and the BrainMap. So if you choose to use BrainMap, this is what things would look like from what you'll see. Okay, so I do have a couple of introduction just to sort of cover where we are. We're talking about the fact that we would like to meta-analyze the statistical parametric images. We don't have as many of those as we do the coordinates, since it is a community standard to extract our coordinates and publish them in the manuscripts. Those can be easily harvested from the papers. But echoing what Tom was saying, please use NeuroVault to upload your images so that we can enhance the image-based meta-analyses that are present in the field. Regardless, we extract those coordinates, and we want to, the angle here is not pleasant.
Here.
Oh, there it is. Okay, perfect. Okay, excellent. All right, so let's just say we have a sample question, right? What brain regions are typically activated in a memory task in schizophrenia patients? Your question can be whatever it is, but this is just an example of what you might be asking. BrainMap.org is where all of the BrainMap software and resources are hosted. We have a forum, we have software, there's various tools. You can look at other AL meta-analytic publications. There is something called a quick author search that if you just want to know if there's a paper in the database, you can use that field right there. We also give you a running tally of how many papers that we have. One of the most notable differences between BrainMap and Neurosynth is the volume of the literature that's archived. That's because BrainMap relies on manual annotations. We actually have human beings who are sitting in a room coding these papers and putting them into the database. In contrast, Neurosynth is much larger because they use automated annotation methods. And there's actually a talk tomorrow in the informatics session about comparing the annotation accuracy that my graduate student is going to be presenting. So I encourage you to go see that. Sidebar. Okay, so 3 main pieces of software in BrainMap. The first one is Scribe. That is to allow you as a user, if you use that quick author search and you go, oh, well, goodness, I have a lot of papers here that I want to put in my meta-analysis, I want them in BrainMap and they're not there, that's what walks you through this manual annotation process and allows you to put the papers in the database if you want to. The other one is Ginger Ale, which allows you to do the meta-analysis itself. That's the Ale method there. And then the last one is Sleuth. And that allows you to search the database according to different metadata fields so that you can sort of hone in on that corpus that you're most interested in. I will say, in regards to the question that was asked during Felix's session, yeah, there's going to be a mismatch in terms of some papers are going to be in database that you want to include in your meta-analysis. Some you're going to find in a PubMed search, Web of Science search, and maybe you'll want them in BrainMap. Maybe you'll want to just simply append those to your meta-analysis file that you create in Ginger Ale, which I'll walk you through. So you can do sort of a hybrid approach. Another solution is to use Roberto Toro's BrainSpell for making some annotations and extracting coordinates. So you can sort of cobble things together using various different meta-analytic resources as you go. But these are the 3 that are sort of constrained within the BrainMap framework. In Scribe, when you go to that web page that's just uniquely for Scribe, we have a download link. And then I strongly encourage people, I know when you get to a website, it's kind of like, where do I click and go as fast as possible, right? Make sure you kind of take a look at some of the resources. We have some sample files. We have some dictionary files that give you a little bit more information about what some of our terminologies refer to. We have a very long manual that walks you through each step of the process. And one thing is that was brought up also in that other question is there's a lot of semantic competition. I can say working memory and you can say working memory and we can agree on that. But when it comes to something like hierarchical classification or processing, then people's terminologies tend to differ a little bit. So we do have a dictionary that we put there that sort of makes people understand what we mean by each of the unique terms that are used to annotate the metadata in BrainMap. And so this is just a little screencast simply because sometimes it helps to have seen some of these things before you actually start to try using them yourself. This is what the scribe interface looks like. Its main critique is, my goodness, it takes a long time to annotate a paper. Yeah, it does. So try doing it for 10 years, and you can see how maybe it can be a little bit frustrating. But it's actually something that has a pretty helpful learning curve. The more you do some of these, the quicker you get. And it's actually, if anyone here is mentoring grad students or undergraduates, it's a fabulous training tool. You get a new student in your lab, and they're like, I want to be a neuroimager. It's like, okay, so you're going to have to read. This gives them an active activity that they can do to assimilate the literature, that you can then go look at their annotations and make sure that they actually grasp the paper that they were reading, and it helps walk them through each step of the process. We have information on our subjects. We have information on the task that was done in terms of the paradigm class, which is the actual task that was done. So that's the NBAC. And then also on the behavioral domain, that's the underlying cognitive construct that you're trying to isolate with the given contrast. And so for NBAC, that would be perhaps working memory. And so each of these panels sort of walks you through all of these different classifications, and at the end of the day, you end up with this lovely little .ent file, and then that gets emailed to the BrainMap team. They review it for accuracy, because again, with manual annotations, there's nothing like the precision and knowledge of a domain expert who's human, who's actually doing the work, right? We're never going to be able to match what a computer can do. But at the same time, there's nothing like a human for making errors, right? Maybe I was just a little distracted. Maybe I didn't really get that point. Maybe I was confused by what this term means versus that term means. So we try to have as many review levels in place as possible to make sure that what is in BrainMap is consistent as possible. Okay, so that's Scribe. And again, I want to point out the particular Excel file that we have on the website that defines what we mean by behavioral domain and what we mean by paradigm class, and gives definitions of each field underneath those classifications, because this is actually a area where you can really debate some of these terms. So we want to at least be clear by what BrainMap means by these terms. Even if your definition, is a little bit different, at least we can be consistent in understanding how things are archived in BrainMap. Okay, so then you have an interest in an area, say working memory, and you want to go and see what's in BrainMap that's under working memory. So that's what Sleuth is for. Again, you download the application. You do need to register for a username and password. This helps us sort of track usage. And again, there's resources available. There's a manual online. And then here we have just a little screencast, again, so that you can see what these things look like, so that if you go to play with it, then it won't look completely unfamiliar. So we developed the interface for Sleuth back when iTunes was brand new, and you could search all these different, you put together your search terms with these little drop-down lists, and we thought we were so super cool. And that's still the interface that we're using, and it works really well that you can construct, I'm interested in healthy participants, or I'm interested in this particular disease category, and I want them doing this paradigm class, or I want them doing that behavioral domain. And then it downloads it to show you how many search results you have, puts it into a workspace, and then you can manually go through, and all of the work that was done in deriving those annotations, you can go through and see all of those metadata files within the workspace, and then the last panel is for the plot panel, so that you can, as Tom said, make sure everything is actually inside the brain. The annotators, they will record the coordinates as listed in the paper. Sometimes authors make a transpose error, and you can clearly tell that X and Y were swapped, and we'll fix that. But for the most part, it's entered as it is. And so every once in a while, you will see a coordinate that's quite outside the brain. But that's your job as a quality control expert to try and identify if that is an issue. And so anyways, that's the last panel there for Sleuth. Now, Sleuth allows you to export your text, export your foci, all of your coordinates, separated by the different experiments, the different contrasts, as a text file. And that's where I'm saying that you can use a hybrid approach where if you go to Sleuth, you find a bunch of studies that match your meta-analytic criteria, and then you go to PubMed and you find a couple others, you can manually append those before running the AL meta-analysis part. So you can sort of make things work for you the way that you want them to. We also, let's see. There we go. Okay, so an example of what this might look like. Tom walked us through what the ALE approach is from sort of a statistical perspective, because that's what Tom does. I'm going to give you the same sort of thing, just represented a little bit different graphically. Let's say you were interested in the paradigm class of face monitor discrimination, and you were looking at healthy participants, and for us that's the context of normal mapping. And so an example is Ken Paller's study from back in 2003. He published memory greater than gender and gender greater than memory. Those were two different experiments. Those are entered separately into BrainMap. And if you were to look across the whole database, you would find 396 experiments of healthy controls that participated in a face discrimination task. That is, we found all these tables of coordinates, we extracted all those coordinates, we put them into BrainMap, and you can look at them in Sleuth, but when you look at them in Sleuth, it's like, well, that doesn't actually help me. So the goal is to look at where they converge. And so again, we're looking at these as centers of probability distributions by using Gaussian modeling. So basically, we're taking that single coordinate point and we're blurring it. That means that when you have all of your foci represented across the brain, you take the Gaussian and you end up with something like this, right? And then when you run a permutation test to determine statistical significance, you end up thresholding the results at some statistical level. And so those references there are sort of the iteration of how the ALE meta-analysis method has evolved from the initial Turkeltaub paper back in 2002. So to do that, what I just walked you through in terms of that series of images, that's the application GingerALE that's embedded in the BrainMap framework. Again, you download it, you get a nice little manual, you walk through all these things. We also have two standard anatomical templates, one in TAILORAC space, one in MNI space. You have to pick one to run your meta-analysis in. If you pick TAILORAC, then all of your MNI coordinates need to be translated to TAILORAC space. If you pick MNI, then vice versa. And that MNI to TAL app is embedded in GingerALE as well, so that we can do that for you. So just quickly looking at the screencast for GingerALE. Yeah, network connections, just hit OK with that. And pick your coordinate file. It'll give you a warning that there are so many coordinates outside the brain. Again, analyses differ. Sometimes you get something that's right on the edge of the mask. Sometimes you get something that's a clear error where you have the brain and then like this coordinate point way out here where that's just clearly an error. But you load things in, and there's some different parameterizations, and Simon's 2016 power study really walked through different statistical thresholding options and why we support the cluster-wise thresholding. But at the end of the day, you end up with a series of files that are all mapped in the manual to telling you what all is what. But you're most interested in taking that last nifty image that's thresholded and looking at it. We support the Mango application for viewing images. You can use your favorite image viewer because these are just standard nifty images. But Mango gives you an option to view your meta-analysis map, which is what you actually want to see when you're done with things. So there's been quite a few different ALE meta-analyses that have been published in the literature. We have a sampling here of some of the different topics. Again, like Simon's terminology, these are topic-based meta-analyses where people from specific domains of interest, they go out and they say, I really want a good assessment of what this particular area of the field has produced. And this can apply also to looking at how these convergence can deviate in particular diseases and disorders. And so with that, I will say thank you, and any questions?
Any questions? So I was just curious, Angie, do a lot of people feed back the information? They should, but I was just wondering if much data comes back into BrainMap in terms
submitted from other labs.
Yeah, or just
external.
Yeah.
Yeah. Again, if you're a PI and you want to do a meta-analysis on a certain topic, you're probably also a PI who gets a lot of, I'm an undergraduate and I want to work in your lab for the summer. And so a lot of people assign, okay, fine, let's go make the annotations for this particular topic that we want to meta-analyze, and it becomes sort of an undergraduate or intro grad student assignment. So there's a steady stream of submissions.
Some nice crowdsourcing.
Yeah, it is, exactly.